Right now, somewhere inside your body, a cell is copying 3 billion letters of genetic code and making a mistake. It happens constantly in trillions of cells because the copying process is never perfect. A healthy cell follows a strict set of instructions. Grow when the body says grow, stop when the body says stop, and if you are too broken to fix, do the honorable thing and self-destruct so you don't cause trouble. Typical cells accumulate between 13 and 44 mutations every single year. Most of these typos land in stretches of DNA that don't really matter. A misspelled word on a page nobody reads, so the cell carries on, functioning normally. Your tissues are constantly weeding out the damaged, the old, and the suspicious. So if the body is this good at cleanup, how does one ordinary, obedient cell manage to survive long enough to cross the line? Cancer is the result of a long, step-by-step failure of the tiny controls inside that one cell. It is a process where the internal rulebook is systematically dismantled. 2 gene families control a cell's life. Proto-oncogenes act as a gas pedal for growth. If mutated, the pedal jams, forcing constant growth. But the cell has brakes, tumor suppressor genes like p53. It senses damage and either pauses the cell or orders self-destruction. You carry 2 copies of these brake genes, one from each parent. Usually, losing one copy isn't enough because the backup still works. You need a second hit, a second mutation, to knock out the safety system entirely. This is why the process often takes decades to unfold. These hits are often written by the world around us. In a smoker's lungs, chemicals grab onto the DNA and bend it out of shape. These chemical warps match the exact spots where the p53 guardian gene is most often broken in lung cancer. When the gas pedal jams and the brakes fail in the same unlucky cell, the threshold of initiation is crossed. The cell has undergone an irreversible change and no longer answers to the body's signals. During the promotion phase, the broken cell begins to divide. 1 becomes 2, 2 become 4, pushing against surrounding healthy tissue. Every division passes on broken genes, providing another chance to pick up new mutations. The cluster undergoes natural selection in miniature. The cells that grow fastest or survive best come to dominate the mass. However, growth eventually hits a roadblock. Once the cluster reaches a certain size, the cells in the center begin to starve and suffocate because they lack oxygen and nutrients. Starving cells release signals that trick nearby blood vessels into growing new branches, plumbing the tumor directly into your circulation. To survive, it also becomes invisible, hiding the flags that attract the immune system. Body patrols walk right past, completely fooled. Securing a private blood supply and hiding from the immune system, the biological engine of the disease is fully assembled and self-sustaining. In the final chapter, progression, some cells pick up mutations that let them break away from their original home. They gain the ability to chew through the surrounding tissue that would normally keep them contained. This is metastasis. Rogue cells slip into the hijacked blood vessels, riding the current to distant parts of the body to set up new colonies. By this stage, the tumor is a chaotic population of cells, each carrying a slightly different set of broken genes. This genetic variety is exactly why the disease is so stubborn and difficult to treat uniformly. The ability to travel and colonize distant organs is the threshold that turns a localized genetic error into a systemic, body-wide crisis. Despite appearing in completely different tissues, most cancers acquire the same set of traits. Biologists call these the hallmarks of cancer, a consistent playbook of biological cheating used to escape the rules of the body. The checklist is the same: growing without being told, ignoring stop signals, refusing to die, and evading the immune system. Understanding this step-by-step nature provides a massive window for intervention. Because these specific mutations often take decades to accumulate, the disease develops slowly. Actionable choices like avoiding smoke and protecting skin from UV radiation directly reduce the environmental hits against your DNA. Meanwhile, early screening catches the process in those quiet chapters before the cells learn to travel. This story is written in DNA. Because we can read the sequence of those steps, we have the biological and medical power to interrupt the progression and change the ending.